far back as 1920, I conceived the idea and the possibility of when the causative agent of malignancy, so-called cancer, would be discovered and found and proven that it would be caused by a microorganism. Of course, reception that I received that far back from the medical profession and scientist was nil. But I kept at the work, and I succeeded in eventually isolating a virus. First, I began sectioning tissues of every known type of malignancy. I sectioned over 20,000 of those, cut them down with a special microtome to very thin. Some of them were only a micron in thickness. I studied those under the microscope, and I eventually built my first high-powered microscope for the purpose of analyzing and checking those sections. The only result that I obtained over all those years was I succeeded in developing a very excellent technique of tissue preparation, but I never found an organism that I could say was the causative agent of malignancy. I went through with this, and then I developed other instruments of greater amplitude of magnification than the old original number one, which was very lacking in resolution beyond about 8,000 or 9,000 times. It was a lens microscope, and all of the air was excluded from the body and replaced with glycerin. The lenses were homogeneous with glycerin all the way through the whole thing, and the result was that in allowing the rays to separate and not cross in the interference band of reflection, such as they do in the ordinary standard tube of the regular research microscope, we held them apart, we separated them, and then brought them back together and picked them up again at a needle point. But, as I say, the resolution of this instrument beyond about anywhere from 9,000 to 10,000 times dropped off decidedly rapidly. We could go up on some occasions, on some preparations, up to 17,000 times, but we didn't consider the resolution anything out of the ordinary beyond about 8,000 or 9,000 or 10,000 times, depending entirely upon the specimen we were examining. With the results that those tissues gained us absolutely nothing. And as I say, I built other microscopes beyond this one. But that instrument we used up until 1931. With that instrument, Arthur Kendall and I, working jointly in the Pasadena General Hospital, succeeded in isolating what we classify the first filterable form of bacteria ever seen. It was isolated from the Bacillus typhosus from cultures that Dr. Kendall brought from his laboratory in Northwestern University in Chicago. And we succeeded definitely in isolating a filterable form of that bacteria. Now, these filterable forms are very minute in size. The smallest of all is the one from the BX, which we isolate from cancer. We have never made a positive claim that that is the causative agent of cancer. But that is the smallest, and it's less than a 1/20th of a micron in dimension and highly motile. We succeeded in isolating this organism, this BX, using a Kendall media. It's known as Kendall media. It's made of a desiccated pig intestine, dehydrated down where it requires about 10 gallons of the original raw gut when it's desiccated, and run through the extractor and dehydrated to make about 1 pound of the original material. Now that is placed in a tube. We use about 4 grams, and then we use 10 to 20 cc's of a tyrode solution. It's a chemical series of salts. Now that is what Kendall used for the isolation or bringing out this Bacillus typhosus in the filterable state, which is published in the Californian Western Medicine in 1931, our joint report on that. Now these organisms, again, before we go farther, they cannot be stained with the aniline or acid dye stains that's used in the ordinary technique and method of staining tissue sections or even bacteria. Now those organisms are so minute and so delicate that they have to be stained with a frequency of light. Now, after we built our first high-powered microscope, we had to build an illuminant unit to be able to see these different types of organisms and different types of tissue also. So we had to build this particular unit. We built a unit which I have a patent on, a special lamp that will produce 2,000 candle power on a needle point of beam where it comes through the condenser. But between this source of illumination and the so-called substage condenser, we have a pair. On the universal microscope we have 4, but we use a pair of rotating wedge-shaped quartz prism. They're circular and they're wedge-shaped. And by rotating those, we bend the beam of light, that's what we term the neutral or core beam of the illuminant unit, at different angles of incidence, so that it strikes the object under examination at different angles of refraction. There we see these particles at a different refractive index, and every one of them is entirely different. For instance, we find that the Bacillus typhosus, in its filterable state, is a beautiful turquoise blue body. We see them swimming through the field. They're highly motile, and incidentally, if the organism, the bacteria, is a motile bacteria, in most instances the virus will be motile or highly motile, and vice versa. So we see these beautiful turquoise blue bodies swimming through the field. With that, we can differentiate absolute and positively, which we have done. We can analyze the blood of a typhoid suspect, the filtered blood serum, or the urine, as men as 10 and 15 days beyond or before the Widal retraction will show that that patient has typhoid, which of course speeds up the diagnostic value of the work. So we place a colon bacillus under this monochromatic light, and there we have a reddish-brown organism that is highly motile also. We place the filterable form of the bacillus of tuberculosis there. We have a jade green, and so on. We place the filterable form, or the BX, that's isolated from our carcinoma, and we have a purplish red one, and so on. We have, from the filterable form of poliomyelitis, we have a coral pink. The value of this particular method and principle is, as stated before, highly valuable in a diagnosis of the case. As we go on with this, it is so important to have a positive diagnosis that is not interfered with by some laboratory faulty technique, staining methods, and whatnot. Now, as we stated before, we'll go back to the point where we contemplated isolating an organism from malignant tissue. The thing is that I positively believed that when the causative agent was found, it would be caused to be found by a microorganism, and not unlikely an organism that we have with us at all times, that is changed by the metabolic shift of the human body itself. That we have proven. I also stated that in one stage it would be not unlikely that it would be found to be in the bloodstream. That was also proven. So as we isolate this organism, we have what we call the BX. It is a very complicated method and technique. The first material that we isolated was from an ulcerated breast mass that I received from the Paradise Valley Hospital in National City. We used and chose an unulcerated breast mass because we'd have less chance of outside contamination. I took those blocks about 4 millimeters square and I placed them in my K media. I incubated them. I had no results. I changed them at different temperatures. I had no results. I was running at that time possibly 400 transfers a day and determining the effect that the argon, neon, and krypton gas, using as a bombardment in a loop, would have upon the growth of pathogenic bacteria. We wanted to determine if it would stimulate or retard the growth. And I was running those things through, as I say, 400 and 500 transfers a day. And there was a vacant tube, a loop that was standing, and this one tube that I had with this malignant tissue in was standing on the bench along there, and it might just as well had a red flag on it. So I picked the thing up and I dropped it in this argon loop. I left it for 24 hours till I was making my transfers the next day, and I looked at it. I saw that it had a cloudiness in it, which it did not have before. Well, I examined it immediately under the microscope, and I found nothing. And I chemically analyzed it, and I found out that it had been ionized by this bombardment of this 5,000 volts in this argon loop. So there's only one counteraction of ionization, that's oxidation. So I put it in a 2-inch water bath, a 2-inch vacuum, and left it in the incubator for 24 hours. And I brought it out, and it had changed again, and I immediately put it under the microscope and began rotating my prisms, and I finally found it alive and teeming with one of the smallest of any of the filterable forms we had yet seen, in a purplish-red refraction under the monochromatic light. They were less than a 20th of a micron in dimensions. Well, that didn't mean a great deal in the way that I carry on my work, because it had been standing around over a period of time and anything might have happened to it. So we started in and run the process through again with the same identical technique with identically the same results. We ran those through many, many times, I think 104 to be exact, and carrying a control with each one at each test. The controls always remained negative, and we always had positive results of attaining our BX. Then we begin our animal experimentation with this. We chose the female albino rat. Now, the method of inoculation which we use is a technique in itself. The animal is kept under quarantine for no less than 10 days, sometimes 12, and its blood checks are made and stool analysis and so on. We want to be absolutely sure that that animal is not susceptible or has any other type of disease. So the animal is shaved and it's kept under a partial anesthesia, because the shock of the hypodermic needle sometimes has effect upon the metabolism of those small animals. We use a long, sterile hypodermic needle that's filled with petroleum sterile jelly, and the inoculation is made no less than 25 or 30 millimeters under the epidermal layer up into the mammary gland. The result is, after about 4 or 5 days, we have a lesion that breaks out in the thyroid area. That we have never been able to determine the actual cause of that lesion that breaks out, but it invariably does. But it heals in a few days, and the tumor starts. And many of those tumors, if they're allowed to grow, will carry a more gram weight than the animal itself. They grow very rapidly on account of the exceedingly high metabolism of the albino rat. So we carried that through over a period of many, many experimental animals, and the result was always the same. Now when we come into the field of microbiology and biochemistry, we find that these organisms can be readily changed to other organisms. We have classified in the whole entire category of so-called pathogenic bacteria 10 individual groups. And we find almost definitely that any organism within that group can be changed to any other organism within its own individual group by the media upon which it is grown. As an example, we can take a pure culture of colon bacillus, and it's, an absolute pure culture that's laboratory tested with no contamination or anything of that sort. And we can change and alter its media 2 parts per million per volume, and in 36 hours we have a typhoid bacillus with every known laboratory check and test, even to the Weedall retraction. So those organisms can be readily changed. But now we'll come to the BX again. The BX will pass through the porosity of the W. Berkfeldt filter, which is known as triple-aught china. It's what the Dresden china is built of. Of course, they are unglazed, but it's a very fine porosity. Now we alter the media slightly of that organism in the tube, and we have another purplish-red organism that we call a BY. Now, this organism is considerably larger than the BX. It will no longer pass the porosity of the W. Bruchfeld. We have to use what's classified as an N for this organism to pass, but it is still in the filterable stage. So, as this goes on through these different stages, it can be readily changed back to a BX and produce the tumor. Now then, we alter the media again. We use a blood serum on this particular type of media instead of the K, and we have another organism that we find there from this identical BY or BX either, that's a monococcoid organism. Now, these organisms, with the proper staining method, can be seen with a good research microscope. And we find them in over 90% of the blood in carcinomatous individuals, in the monocytes only. And the monocytes are the only of the white cells which have no digestive power, or what we call phagocytosis. Now we alter the media once again, and we place it this time on a hard-based media built of agar and asparagus, ground green asparagus. Tomato will do also, but the asparagus is more potent. There we have a fungoid organism with all the branching forms. It comes in, and we have classified it as Cryptomyces pleomorphia. It's a pleomorphic fungi. Now, it comes in through from the hyphae and on through up into the spores and the ascospores and all its branching forms. And as the ascospores, when they shed or come out from the buds of the spores themselves, are for a very short time highly motile. That is, we presume they are. It may be a Browningian reaction as far as we're concerned on that, because it isn't a true motility. They just have a movement. Now, we allow that particular culture to stand. We can throw that back at any moment into a BX in 36 or 48 hours and produce a BX. But we allow that culture to stand as what we term a dormant stalk culture for the period of metastases. Now, it is very seldom that we have a true case of metastases from the removal of the initial mass of one portion of the anatomy, so where it reoccurs and shows up in another. Less than a year. We allow that particular culture to stand for the period of 1 year as a dormant stalk culture. We plant it back on its own asparagus base media. We allow it to incubate and grow. We do not have a Cryptomyces pleomorphia. We do not have a monococcoid organism, which we find in the bloodstream of carcinomatous patients. We do not have a BY, or we do not have a BX, but we have, from an organism that was isolated initially from an ulcerated human breast mass, we have a colon bacillus that will pass every known reaction of laboratory technique. It is important to record these things, which of course we have, but the thing of it is important that the metabolism of the individual can readily change a non-pathogenic organism into a pathogenic bug that will absolutely produce a disease. There has been much controversy as to the different classifications and qualifications of malignancy. Many people believe that the sarcoma will turn into a carcinoma. We have found in some experimental animals that that is true, but nothing that we can place as absolute. Now, the different types, we have done very little work on the sarcoma because we do not associate that as a malignant tumor in comparison with the work we have done with the true carcinoma. And we have a type that is a bone tumor called shroudinoma. Now, that is a true malignancy, but it's a malignancy of the bone joints and the bone marrow. That affects the type of your white blood cells greatly. Many of our types of anemia will come from that, and leukemia, and so on. But we cannot absolutely say that that is a malignant tumor because we have not been able to isolate a BX from it. We must be able, with the organism that we isolate from a certain culture to be able to place that into the experimental animal and produce the identical tumor, which on removal of that tumor surgically from the experimental animal, recover again that identical organism, which we have done in over 700 cases of experimental animals. As we place the BX in the tissues of the animal, we allow that tumor to grow, and it's removed surgically. The animal is very seldom ever harmed outside of the removal of the tumor, and we section that tumor, and we also study its pathology, and we also check and produce a BX. That is what's known as Koch's postulate, where we inoculate the animal, we recover from the animal the organism that we have placed in it in its true identical form, and it can go on through as many transplants as desired. But when we can take an organism that is isolated from a human being and plant it into an experimental animal and recover all the true pathology of neoplastic tissue from that tumor in the animal, and again recover from that tumor the BX that can be placed into another animal and produce the tumor, why we consider the cycle complete of our series. For many years, laboratory workers and technicians have been producing tumors in animals, especially rats and mice. But they are not inoculating the virus. They are inoculating the cells from the ground-up tumor, which does not signify anything in our work at all, because that's been done over a period of years. It's interesting, but it does not show us anything. What we want to know is the end results of the accomplishment of the placing of this organism in the tissue of the animal. Many people have attempted to use guinea pigs for work on malignancy. Well, we have had no luck with it whatsoever. Guinea pigs are very susceptible to your tuberculosis and different diseases of that sort, but we have never been able to produce a true tumor in a guinea pig. They have sometimes a spontaneous tumor that resembles a malignant growth, but it will not show in true pathology the end results. So that is what we wish. The same as we isolate the filtrable form of the poliomyelitis. As I say, we have a coral pink object there, which is non-motile. Now, I worked a long time, and Rosenau and I worked together, Dr. E.C. Rosenau, the head of the research department of bacteriology in Mayo's. He worked for many years of the isolation of this organism, and he worked with a brain emulsion. I found the organism in the spinal fluid, the true virus. We attempted to use different types of animals, chimpanzees, monkeys, and so on, and we found that the only true susceptible experimental animal to the adaptation of the virus of polio was the albino rabbit. With those results, we produced all the true paralysis in the animals that had all the symptoms and the whole thing. The organism that's isolated, Rosenau still calls it the virus of the Streptococcus of poliomyelitis, which it may be that I will not contradict, because he has worked for many years on a Streptococcus organism that is isolated from the brain emulsion of patients susceptible to polio. But we do not find that the other animals will take it, only the albino rabbit with all the true symptoms. And those organisms can be changed around, as we stated before, by the metabolism. There's been much controversy about the different types and the association of different types of tumors, malignant and what we classify as non-malignant tumors, such as the sarcoma and so on. We have done little or no work on the true sarcoma as to its relative association with the true malignant tumors. We have worked some with the sarcoma of the barred rock chicken, which is known as a Rausch sarcoma. It is an interesting organism. It is not truly a filtrable form. It can be seen readily with a research microscope on sections of the tumor. But it is interesting in one particular phase that it is a transmissible disease. It can be transmitted from one chicken to the other, and incidentally, the only fowl known to science that is susceptible to this Rausch sarcoma is the barred rock chicken. They have it when it gets in a flock, and they all have it. The experimental work that we did with that was attained most of our tumors from the zoo hospital in San Diego at one time, back in around 28 or 29, somewhere in there. They gave me some of those tumors, and I experimented with them some. We never attempted to transmit them back by inoculation or transplanting of any other way into the chickens, but we did do some work on them, and we can see by the section it on a real high power of a good research microscope with a proper staining method. Incidentally, the staining method that we use on that is very similar to the staining method that we use as a flagellate stain. Now, I developed a number of years ago a stain for flagellated organisms, such as the B. coli and the typhoid and many others, that is the most outstanding of any of the stains that I've tried to use, and I've tested and checked through all the flagellate stains that was ever built. And this particular stain is made from an ink that we produce from alfalfa hay. The material is taken in a very small test tube and agitated slowly with 1 or 2 cc's of mercury. And we presume that this mercury has the same effect as a tinning of a wire when you wish to solder it. And you take this ink that's distilled, or extracted rather, from the alfalfa hay and put in there and move it back and forth very slowly in an agitation, not shaking it or anything of that sort. And there you will put your material out. Of course, they are dead. They're no longer motile because the stage will kill them. And there we see the beautiful flagellates. We developed this particular stain for this reason, because in many cases of typhoid, we have a high contamination of colon bacillus. And by staining the masked material, we can take, and under the microscope, we can determine by our counting chamber the number of coli and the number of typhoid that we have in a certain series of squares in the counting chamber. Now, it's just the same as the staining of these organisms. It's a delicate technique that we stain them with the monochromatic light. And certain blood organisms we can stain with a monochromatic light. Even the larger ones that cannot be stained with any acid or aniline dye stains. So as we go through with this thing on a long period of research, we find that the staining method in certain phases of this is of vital importance. Now, as stated before, we cannot see or stain the virus, because the particles of the virus are much more minute than the chemical granules of the stain that we attempt to stain them with. So as it's the old saying, the mouse, he cannot swallow the elephant. So as we go on with these different types of tumors, you take, for instance, the epithelioma, which is a true type of skin cancer. We have isolated from epitheliomas not numerous times, but a few times, our identical BX. We have isolated it from the tumors of the pyloric orifice, the stomach, and so on. We get the identical BX from the true cancer, anything that we can get true pathology from on our sectioning and staining method under the regular microscope. We can isolate from that particular organism a BX almost 100%. As we examine the blood of a carcinomatous individual, we find little or no real action. We know that in the monocytes of the white cells of the blood, we find these small monococcoid granular organisms. But we have as yet to find any effect on the red cells that malignancy has. The white cells will come to any combat, to any infected area, and they will attempt to devour the virus, which they do to an extent. But the growth of malignancy is so much more rapid than they can compensate with that it's just almost difficult. And the monocytes, as we stated before, are the only ones. We've run our blood counts through our hemocytometer, through our actual blood counts of the cells before and after the treatment of these cases, or before and after the removal of the tumor. And we find a change in the blood count. The normal count of cells, as any laboratory technician knows, for the average individual is around about 5 million per cubic millimeter. Well, that will rise or lower. And in case of a severe case of malignancy of the liver or the lung, the count will drop down to 3 to 3.5 million on the individual, and a female much more. The white count will generally rise, but we count those in a hemocytometer. We use an ordinary staining system, and we use a pipette that is calibrated for the red cells and the white cells on a certain amount of dilution. And it is a waste of time, of course, to explain this method and principle of this thing, because any laboratory technician does it hundreds of times a day. But as we run our counts, we find that it is a general lowering of the count in the case of malignancy over normal, and generally a rise in the white count of the blood. The lymphatic system can absorb a certain amount of toxins that's thrown off, and beyond that, why, then it is produced and it comes back again into the bloodstream. But as far as any deterioration that we have ever seen, they claim in some cases of myelogenous leukemia. They call that cancer of the blood. Well, possibly so. But it's a deterioration of the blood, especially the white cells. The count rises up into triple and quadruple their normal amount, and the red count generally deteriorates down to as low as 2 million. But it does not signify anything, because we find in that particular type of malignancy of the blood, as it's termed, myelogenous leukemia, we find the BAS cells, the eosinophil cells, the polymorphonuclears, and so on, their morphology has changed greatly. But it is only the disease that has changed the blood and not the blood that has changed the disease. We consider, as this particular stage, as we call the monococcoid form of this VX, enters into the bloodstream through the lymphatic system, it's carried through basically by the hemoglobin and the blood serum, which we classify as hemoglobin, of course. There in reality is no such a thing as a red cell or a white cell. They're merely classified as that particular type to differentiate them. So in the field of hematology, we go through and we study this blood in all stages and forms. And as stated before, we find that the only type that we can actually distinguish that is associated with malignancy is found in the monocytes of the blood. And as the monocytes, as stated before, are the only of the white cells which have no phagocytosis. There is much controversy as to the pH of the individual and also of the test tube in a laboratory. Now, we have found, experimenting with these different types of so-called malignant bacteria, that some are susceptible to an acid and some to a different types of base. They're positive or negative, alkaline or acid. Some bacteria are susceptible to the acid type and some to the reverse. But we believe that an individual that has an absolute neutral metabolism is susceptible to no disease, because we can take, and we have repeated this experiment not once but hundreds of times in the laboratory with very careful and positive technique, that we place 3 test tubes, 1 acid, 1 alkaline. The center tube is a neutral pH. There is not an organism in the whole category of so-called pathogens will grow on that neutral pH. And as I say, it depends entirely upon the acidity or alkalinity of the organism of which we wish to grow. To make a pure culture of these organisms, especially the virus, it is a very technical, long drawn-out process. Some of them are so-called anaerobic, and some of them are oxaerobic. Some of them require oxygen, such as the BX requires oxygen in one stage of its form to grow. To take the poliomyelitis is strictly anaerobic. That has to be developed in a media that is absolutely free from air. The best media that we have found so far is what's known as a chick infusion broth. We take chicks just unhatched, use the brain and make an emulsion, and that we grow the polio on that because it is associated and has almost the same affinity as the spinal fluid of a human being, with results of that. As stated before, we believe that if the metabolism of the human body is absolutely normal and a neutral pH, it is susceptible to no disease.
Doc, would you mention the polarity that you encountered with these virus under the microscope?
Well, some of these particular organisms, not only the virus, but many of the pathogenic organisms have an individual polarity. Now, we have tested those out, and we find that the positive organism will not associate with the negative organism. We show that where we use a 1/1,000,000th of a volt under the slide in our hanging drop, and some of the organisms will come to one side and some the other. Now, for instance, in the case we get back to the BX, that is a bipolar organism. It is susceptible, partial of it, to one pole and some to the other. We show the negative pole, and many of the motile organisms will come to that pole. We show the positive pole, and the rest of them that are motile and swimming will go to the other pole. Well, we go in with the micromanipulator, with our micropipette, and we draw off from this drop the negative side. We transmit that into the animal, and we have no malignant growth. We draw off the positive side, we put that into the experimental animal, and we have no growth. But if we take the material from both the positive and negative pole, there we produce the true malignancy of neoplastic tissue.
Now, the thing that I would like you to explain briefly is somewhat of the technique that was accomplished in making the photomicrographs that appeared in the Journal of the Franklin Institute. First, I wish you would tell us a little bit about the sectioning of the tetanus bacteria.
Well, this particular organism that Mr. Crane speaks of, this is a spore of a tetanus. This was not sectioned. This was merely the spore of the bacteria in an unfilterable state that was placed between a cover slip and the slide and then photographed. That was photographed over the big universal microscope, the one that is appeared in there with a very high magnification, and also the Bacillus typhosus was photographed over the large microscope. Those was stained. What staining that they have was stained with the monochromatic beam of light. There was no acid or aniline dye stains used upon them whatsoever. And that way we succeeded in getting a photograph. Now, that was taken on the stop motion. I was photographing the life cycle and life development, which we have photographed of practically all the pathogenic bacteria from the cradle to the grave, as it were, and on stop motion, the same as the pictures of the blooming of flowers are made. With the device that we had at that time in the laboratory, it'll photograph anywhere from 32 in 1 second down to 5 hours duration between each exposure. And it's grown with an electrically heated incubating stage under the microscope, and it is carried on through into the film, where we worked 3 cameras simultaneously over that, one still, one motion, and one that was used as an emergency camera. And those were photographed, some of them over a universal and some over the number 3 microscope. Mr. Crane wanted me to speak about the particular typhoid that is shown in that article that was published in the Franklin Institute, was also published in Smithsonian's magazine also. That is grown on K media, and we photographed that one in particular because it's a very, very rare case that we find a vacuole or a virus in either end of the bacteria. That was why that one was particularly photographed. We have photographs of others, but that one there, we made a special effort to photograph that. Now, this K-media, as stated before, has the faculty of transforming these organisms into what we term the transitional state. So they will shed these virus, and then when we filter them, only the virus will come through the porcelain of the filter. And that is just in the transitional state, that particular Bacillus typhosus. It does not show the flagellate on there because the flagella will not show with the monochromatic light to any degree of intensity whatsoever, not enough to photograph. So that was how that one was made. That was merely in a fluid mount, and as it slowed down its motion, getting ready to shed the virus, we photographed it. And that is where that came from there. Now, the so-called incubation period of these pathogenic organisms, as long as we have a little tape, we might as well finish this out here, this part of it. These so-called pathogenic organisms and their incubation period, we consider not an incubation period at all, but merely a cycle of reversion, waiting until that organism becomes in its transitional state. Now, those organisms that we find associated with a disease, we do not believe cause the disease in the form in which we find them associated with the disease, but they are caused to be in that particular form by the disease. Now, as an example, as I've stated many times before, we will take as an example the Bacillus of tuberculosis, which has an extremely long so-called incubation period. We inoculate that particular pure culture, laboratory tested of a Bacillus of tuberculosis, into our guinea pig, and over a period of weeks we develop in the lymphatic chain of that animal the symptoms of the tubercles from which we can again recover the tuberculosis. But if we inoculate the filterable form of that Bacillus tuberculosis, which heretofore has been known as the poison molecule of on, it is merely the chemical constituents of the organism, we produce the disease in 36 to 48 hours. We have eliminated 9 stages of fungi from the bacillus form on up until it becomes a filterable virus or the premoidal cell. We sincerely believe it is the chemical constituents of these organisms that produce the disease, their predominant chemical factor, which is the virus or the premoidal cell. So, we inoculate the premoidal cell into the animal and we produce the disease in 1/10 the actual time that we produce by the inoculation of the organisms. And we sincerely believe, as stated before, that it is the chemical reaction of those viruses upon the unbalanced cell metabolism that produces a disease, because we can produce in most instances all the symptoms of the disease chemically with no bacteria. Now, as we take again the bacillus of tetanus, we know, of course, all pathologists, all bacteriologists knows that it's caused by an infection, or it causes an infection. It's an organism that produces blood poisoning and lockjaw. So it's associated with that particular disease. It comes to any type of infection, same as your staphylococcus and streptococcus. They produce many different types of infection, but in most instances they are in a form in which we find them by the disease itself. They're carried through by contaminations of air or whatnot. They are not as important, of course, as tetanus. Any infection that is produced by anything that is ionized or by iron will produce a tetanus infection, such as stepping on a rusty nail or things of that nature. It's very prevalent in certain countries, of course, and it is transmissible in certain cases, especially with open stores and infection where they have leprosy and all of that different type. But your streptococcus and staphylococcus are, in most cases, you find them with any infection. Now, as stated before, Dr. E.C. Rosenau and I have worked together for a number of years, and he stresses practically everything on streptococcus. Now, as we isolated the virus, and in his publications, he still calls it the streptococcus of poliomyelitis. And they get along on those certain lines, and there they stick, and they follow through, and that is their end. All of these different types of diseases that come through, why, they are caused, many of them, by the disease in question, with exceptions of your parasitic diseases and things of that sort. We have done over a period of years a great deal of work on the animal parasites of man and the different types of blood diseases. And your animal parasites of man, such as your types of trypanosomes of African sleeping sickness and your Mycophilaria bancrofti of elephantitis and things of that sort, those are both transmitted by the tsetse fly. And there are so many of these different types of ailments appertaining to human disease and animals that they're just absolutely too numerous to mention. And as we go through with them and we study them and photograph them, study their life cycle, their development, we know their incubation period or so-called incubation period of them all. So that is something that will have to be worked out farther by other technicians and other workers besides myself. Mr. Crane wanted me to make a few remarks on leprosy. I worked over a period of time with Dr. Linwood Walker that was connected with the Hooper Foundation in San Francisco. That's a satellite, might be, from the University of Southern California in Berkeley. And he was very much interested in the association of the different types of leprosy. Now, we have three different types of leprosy. We have the soil leprosy, the rat leprosy, and the human leprosy. And there was much controversy as to the difference in the morphology of those organisms, which in their staining method they were different, and in their morphology through the microscope they were different. But we run through a series of those organisms in the filterable state, and we found definitely that the rat, the soil, and the human leprosy in the filterable stage was identical with one another. We never attempted to transmit any of these organisms. Dr. Walker had them there after we isolated them, but we run them through and we found out over a period of a long series of research that they were identical in the filterable state. It's a very interesting organism, and we have devitalized under electrical application the leprosy, the organism, both in the rod and the filterable state. It is in the human form. Now, we have gone through, as I say, I have worked at this work for over 50 years. I have worked sincerely and honestly. I have never attempted in any way to fool myself. It's a very easy thing to do, to fool oneself when he works a long time on a particular project and he looks hours and hours a day through the tube of a microscope. He can imagine a great many things that he does not see. But the work is encouraging, and it's something that can be carried on with the improvement and the advancement of scientific instruments that we have at the present time. It will carry us farther into the field of endeavor. And we hope sincerely that what little we have done thus far will be a stepping stone, an endeavor for the great field of workers and scientists in the future.